Hi, my name is Romy Amaro from UC San Diego, and I'm here today to tell you about our Gordon Bell paper entitled AI-Driven Multiscale Simulations Illuminate Dynamics of the SARS-CoV-2 Spike Protein. I'm just one person of an incredible 28-member team spanning the entire United States and part of the UK, as well as 4 of the most amazing supercomputing sites on the planet. I'm so proud of all that this team has accomplished. So some of you may have already read about our work in a New York Times article that went to press the day after we submitted the Gordon Bell paper. And I was so impressed by how they portrayed our beautiful imagery, as well as for the opportunity to bring HPC and molecular simulation into the living room of millions of people worldwide. And what this article couldn't do, or didn't do, was actually discuss the scientific and computing technical challenges that we faced when trying to carry out these simulations. And so that's what we're going to try to do today, is to describe at a very high level what we've done with this submission and give you the science behind the pictures. I like to think of our methods as a computational microscope, because what we're able to do is to combine different kinds of experimental data into one cohesive model to develop an atomically detailed physical model of a system. And we approximate that system down to its many atoms. We then define a potential function which describes the interactions of the particles or the atoms in our system. And then all we're doing is integrating Newton's equation of motion over time. And we perform this integration on the fastest and best supercomputers that we have available, such as the Frontera machine and Summit. And we perform this integration on those machines millions and billions and trillions of time steps, and we're able to build up, at the end of the day, a dynamical understanding of our system. And why that's so important is that one of the things that we cannot see with the experiment is atomic scale dynamics. So this approach really has the power to extend and augment experimental data sets beyond where they can currently reach. But no matter how powerful these microscopes are, they are still challenged. And a central challenge that we are concerned with is how to really bridge molecular detail into subcellular and cellular scale scenes. And so what you're going to hear about today is how we used, for example, weighted ensemble simulations in order to address the time scale challenges, as well as AI-driven workflows in order to sort of cross those key capability gaps where otherwise it would be very difficult to bring atomic scale resolution into these longer and more complex scenes. This timeline describes the highlights of the science and computing milestones for our submission. And to me, really what I think it emphasizes the most is readiness. So in mid-February, the very sort of first event that happened that was really critical was the deposition of the first cryo-EM images of the viral spike protein. That same week, we had just published a landmark study of the influenza virus. And so the pivot for my group was immediate and natural. That same day that the structures were deposited, I contacted Dan Stanzione at TAC and asked him about getting access to Frontera so that we could begin to explore this viral protein without delay. Dan responded immediately and was extremely supportive of our efforts. And since then, that really kicked off the whole end-to-end view that you're looking at here. And the other thing that I still find hard to grasp really is how far we've come in so short a time. The work represented in this timeline represents what just even a year ago, nobody could have ever imagined we would be able to complete. But faced with this challenge, this incredible team, and the resources made available through a variety of efforts, we were able to meet the challenge that COVID-19 brought us. So what you're going to be hearing about predominantly is the so-called spike protein. And why we care about this protein is because it sits on the outside of the virus and is the first point of contact that the virus has with the human cell. So it's really its main infection machinery. And what I'm showing on the left is a movie that it's a trajectory actually of those atomic level dynamics of all the atoms in the system. And so a couple of things I want to bring your attention to, those little balls that are hanging off of the the structure, they look like Christmas tree ornaments. Those are the glycans. And one of the main challenges here is that experimentalists cannot see what those glycans look like. So this is a place where HPC really has a lot to give. And that's why it's one of the sort of big science outcomes of this effort. I also want to bring your attention to something called the receptor binding domain, which is this little blue bit at the top of the spike. That bit of the spike turns out to be very important for reasons I'm going to talk about next. In order to get a real understanding of the infection process, we also need to simulate how the spike protein actually latches onto the host cell. To do this, we also simulated the ACE2 receptor, shown here in yellow, which makes a critical interaction with the spike protein. This interaction is like a handshake between two people, and this contact initiates the cascade of events that cause infection. One thing I want to bring your attention to again is that light blue bit in the middle, the so-called receptor binding domain. This handshake with the host cell can only occur when that blue bit is in the open or attack conformation. The process of receptor binding domain opening is critical to the infection process and something we will keep coming back to. The other major challenge, in fact the single biggest obstacle to simulating the entire viral particle, is getting the membrane right. And this is because of the difference in curvature between the inner and outer leaflets of the membrane, which makes all sorts of interesting and challenging things happen. So what is required is an intensive iterative modeling process, shown here. I won't go into its detail, but we go through a series of essentially patching different holes and deformations in order to make that viral particle membrane both biologically realistic as well as accurate enough physically for numerical simulation. And this is what we're left with at the end of the day. This is the fully reconstructed 305 million atom SARS-CoV-2 viral envelope, which has the correct spatial patterning and density of the spike proteins on its surface. Here you can see all of their atoms. And this is so striking and amazing for the world to see because we are giving people never before seen intimate views of this virus with resolution that is impossible to achieve experimentally. And again, why we care about this is because if we want to understand how the virus infects the host cell, if we want to be able to design antibodies and new drugs to block and cure infection, if we want to be able to design new therapeutics, this information at this very fine resolution at the atomic level is required. With that, I'll hand it off to John Stone, who will talk all about the software and HPC innovations that made this work possible.
NAMD and VMD are the flagship molecular modeling applications developed by our laboratory at University of Illinois. VMD allows researchers to combine multiple modalities of experimental structure information from X-ray crystallography, cryo-electron microscopy, and homology modeling to build and analyze atomic detail models of large biomolecular systems, such as viruses. Once the structures have been prepared, then they can be simulated on parallel computers using NAMD. NAMD is designed to provide both strong parallel scaling for a single simulation and also to accelerate ensemble sampling and replica exchange approaches. Both programs are unique in their support for large-scale parallel computing platforms and the size of the biomolecular systems that they can model. NAMD and VMD were partially prepared for the demands of the SARS-CoV-2 project as a result of our prior large virus simulation collaborations. With researchers all over the world working from home with reduced access to powerful workstations, we've been engaging in a very responsive development cadence. We are providing unusually early access to developmental versions of NAMD and VMD to get recent advances into the hands of the research community as quickly as possible. Some example innovations that had an impact on this project included significantly expanded support for biomolecular systems of up to 1 billion atoms. We've also added support for hardware-native, low-level message passing systems such as PAMI on Summit and UCX on Frontera. This gives NAMD about a 20% performance boost over portable MPI-based communication. Working with NVIDIA engineers, we've incorporated optimizations that sped up the non-bounded force kernel by up to 20%, and as a result of a new collaboration with Intel, we've incorporated a new AVX512 non-bounded force kernel that yields a 1.8x overall speedup for NAMD running on TAC Frontera. Our latest NAMD advance is a new GPU resident compute mode that increases performance for single-node, ensemble, and replica exchange simulations. The new mode gives us a factor of 2 speedup on a single Volta GPU. Just this week, we've enabled multi-GPU support to further speed up simulation of replicates on tightly coupled GPU-dense node architectures like Summit. All of our CPU and GPU kernel optimizations benefit not only runs on supercomputers, but also departmental clusters and workstations as well. Looking at the NAMD performance results, we've shown strong scaling of the 8.5 million atom spike plotted in the top 3 traces of the scaling chart. Running on 1,024 nodes, there are only about 8,000 atoms per node, which is a tiny amount of work for nodes with large CPU core counts or multiple GPUs. The 305 million atom full virion scaling result is shown for up to 4,096 nodes on Summit. It's worth noting that at this scale, there are only about 12,000 atoms per GPU. This is a regime where some of the NAMD force kernels run into significant work starvation. Despite this, NAMD still manages to achieve 51% scaling efficiency and about 3 petaflops. Taking into account the inability to hold off on production science runs to rewrite the affected kernels, we are happy with this result. To ensure that NAMD and VMD perform well on current and next generation platforms, we maintain long-term collaborations with supercomputing centers and hardware vendors to allow us to take advantage of new hardware and software architectures and programming models. If it wasn't for our participation in software readiness collaborations for years in advance, it wouldn't be possible for us to have NAMD and VMD in a usable state when these next generation systems are deployed. Finally, we are proactive in publishing the resulting algorithmic, methodological, and technological advances on an ongoing basis. Now that you've heard about the contributions of NAMD and VMD, Lillian Chong will tell you how we used advanced sampling methods to reach effective performance far beyond what is possible with brute force computation.
Thanks, John. I am going to tell you about how we simulated the opening of the spike protein that occurs before it can latch onto cells during infection. Standard MD simulations of spike opening could take years due to the large free energy barrier between the closed and open states. In this simplified diagram on the right, I show the progress of a set of simulations indicated by spheres towards the target state. Due to the large free energy barrier, the simulations will spend most of their time in the initial state, waiting around for a lucky transition to make it over the barrier. Fortunately, the transition itself is relatively fast. On the left is a movie of a lucky transition. In distributed computing projects such as Folding at Home, thousands of simulations are run starting from the initial state to increase the chances of capturing such lucky transitions. However, these simulations are still spending much of their time in the initial state and not enough time on the interesting functional transitions between the initial and target states. So how can we be even smarter about capturing the interesting transitions? One way of doing this is to use the weighted ensemble strategy. The transition you just watched was one we captured using this strategy. In this strategy, we divide a progress coordinate towards the target state into bins. We then run many short trajectories in parallel. At fixed time intervals, we enrich for success by replicating trajectories that have made transitions to empty bins, and we save computing power by terminating trajectories that have not made transitions. The elegant part of this strategy is that the trajectories are weighted, as indicated by the sizes of the spheres, and we track these spheres in a rigorous way such that no bias is introduced into the dynamics. By minimizing the waiting time in the initial state, weighted ensemble MD generates unbiased and continuous pathways with orders of magnitude less computing power than standard MD. This efficiency gets even better for slower processes as the efficiency scales exponentially with the free energy barrier. Finally, the resulting pathways are the mechanism, enabling us to directly analyze how the functional transitions occur, including transient states that are too fleeting for experiments. Our weighted ensemble simulations were run with my lab's open source WESPA software, which is highly scalable out to a large number of GPUs as well as CPUs. The dynamics were propagated with the Amber software using 100 GPUs on Longhorn. And our simulation is a crowning achievement, generating an ensemble of pathways for a long timescale process with a large system of 600,000 atoms. Our weighted ensemble MD generated more than 1,000 pathways in 23 days. Assuming that the timescale for spike opening is somewhere in the range of microseconds to milliseconds, standard MD simulations would require 10 months to 8 years to generate just 100 pathways. In other words, weighted ensemble MD is 13 times to 127 times more efficient than standard MD. I will now hand off to Arvind, who will tell you about how these pathways were used for his DeepDriveMD efforts.
Thank you, Lillian. One of the things that we very quickly realized with these weighted ensemble MD simulations was the fact that this was extremely large. For the spike trajectories, it was over 200 terabytes of data. Obviously, it would be humanly impossible to look at all of these data frames and understand what sort of conformational changes are going on. In order to do that, we decided to use machine learning, specifically deep learning approaches, to characterize the conformational transitions. We take the inputs from these simulations as coordinates, contact maps, as well as other features computed from these trajectories, and process through them using our deep learning ideas. The objective of the deep learning idea is twofold. One is to build a low-dimensional representation of this data such that we can identify all of the conformational transitions that are sampled from these trajectories. Note that this has to be physically interpretable, which means that we need to understand what sort of conformational transitions exist, and we can measure that using the root mean squared deviation, which is a measure of structural similarity across different trajectories. Furthermore, we also need to cluster the data so we can identify what sort of states have been sampled within these simulations. We take these outputs from these simulations and try to understand what are the interesting conformations from them, such that if we identify some sort of motions within the receptor binding domain, for example, this is a motion where we see the opening of the receptor binding domain, we continue running such simulations. If not, we stop simulations that are not that interesting. We have shown that this technique works very well with protein folding data. You can look at the papers that we have published on this area, as well as the website that we have pointed to here. The traditional approach of analyzing this data was using autoencoders. These autoencoders took the trajectory data and built an image representation, specifically a 2D image representation, which we run convolutional filters and encode that space using a low-dimensional manifold. Obviously, this brings about two main challenges, one being the fact that we are using sparse matrices as inputs from these trajectories, and as well as the fact that for the spike protein, this amounts to about 1 billion parameters for training across the trajectories. This inherently means that it's very hard to train on such large trajectories. Therefore, we came up with a solution which allowed us to directly work with the 3D coordinates that are generated by these simulations. So these XYZ coordinates are usually represented as point cloud representations, and we used a similar encoder-decoder architecture that we were using before, but with one significant change, which was that we added an additional adversarial layer for basically training these autoencoders in such a way that they become more robust to input noise. Obviously, this allows us to quantify what constitutes a good reconstruction based on the data that we have seen, as well as what constitutes a bad reconstruction, given the fact that all of these approximations might not be the same. This innovation really allowed us to work with a far few number of parameters. In fact, it's only about 10 million parameters for the spike protein, and allows us to train on such large data sets without having to worry too much about computational performance. Indeed, this gives rise to good computational performance, as you can observe, with respect to the variational autoencoder, which takes quadratic amount of memory as we scale up on the larger spike systems, whereas the adversarial autoencoder varies linearly in terms of its memory consumption. The second important aspect is the fact that the training time per epoch for the adversarial autoencoders remains almost constant throughout the scale of these proteins that we have simulated. At the same time, the variational autoencoder varies quadratically. This gives rise to good performance in terms of scaling onto such a large system, especially given the fact that we need to consider the 305 million atom system that we're also working with. In terms of understanding what does this autoencoder represent for these spike conformational transitions, the largest conformational change that we observe is about 10.5 angstroms from the native state, which represents the closed state of the protein. Obviously, when you look at all of these trajectories, we can map them onto this neat space, three-dimensional representation of what the adversarial autoencoder has learned, and also map out large-scale conformational transitions onto the same space where we can look at some of these receptor binding domain motions. Obviously, we have hidden all the glycans and everything for clarity, but you can see that the learned space actually encodes all of these conformational transitions. The next set of this data we started looking at allowed us to visualize some of these outliers when it came to look at both the SPICE and the ACE2 receptor simulations. When we look at the SPICE and ACE2 receptor simulations, we do identify a bunch of outliers in the same space, but with a much smaller degree of freedom, where you can observe that the overall RMST varies between 3 and 5 angstroms. Obviously, this shows significant motions in certain parts of the protein, including the spike stalk region, which is what we wanted to sample a lot more. The second time we also looked at these motions, we were able to capture even more of these conformational changes, which allowed us to determine that these are true outliers from the data. Obviously, when we use these outliers in sample, we observed that within 12% of the time that we used to simulate, we were able to capture over 25% more conformations, which allowed us to quantify the speed up in terms of sampling efficiency to about 8 times what was originally done. Given the fact that we are able to scale it on to 1024 nodes of Summit with the Ensemble Toolkit workflow that we have developed, this allows us to confidently promote the idea that such AI-driven simulations can have a broad impact in terms of how simulations are run. With that, I would like to thank you, and I'll pass the baton back to Romy, who will talk to you a little bit about the scientific outcomes of our study.
Thanks, Arvind. In this last segment of the talk, I want to just touch on the highlights in terms of the science as well as the impacts for HPC. And I'm going to start with what's turned into an iconic image, which is of the spike protein. On the left, what I'm showing is what the spike protein was thought to look like before simulations. And on the right, you can see how different it actually looks when one simulates and includes those glycans that experiments cannot see. And what's really key here is not only knowing where that protective shield is, but also where it's not. Because in these spots that are open, where the protein is actually exposed, those are the vulnerable parts of the virus and of the spike protein, where antibodies bind and where possible new drugs and therapeutics could be designed to also prevent or cure infection. Our simulations also showed why the spike opens and closes. And what you can see here, there's 2 different modes. We showed that the spike has a so-called defending mode, where it buries that receptor binding domain down underneath the glycan shield. And it also has an attacking mode. When the RBD is in that open conformation, that really important part of the spike protein that needs to make contact with the host cell is exposed. Beyond shielding, our work also established a structural role for the glycans. As you can see here, there are 2 glycans in particular that almost act like a kickstand on a bicycle, propping up this receptor binding domain in the open conformation. These basically are locking and loading the spike for infection. Weighted ensemble simulations at scales never before achieved show us how the spike actually opens. And what's so important about that is knowing the pathway of opening presents new opportunities for drug design. One can start to imagine finding molecules that will trap the spike in particular conformations to halt the infection process. We then extended this work and used artificial intelligence-based workflows as a multiscale glue to bridge key capability gaps in terms of the spike protein at the next level of complexity, as it forms that critical handshake interaction with ACE2. What was really key here was having the AI learn information at the smaller scales in order to drive the conformational sampling of the spike within the more complex scene. This enabled a speed-up factor of more than 8x relative to standard approaches. Our submission culminated with the all-atom simulation of the SARS-CoV-2 viral envelope. I'm showing you here just a small snapshot of the dynamics we've computed. What's so exciting about this is how it unlocks entirely new dimensions for studying viral infection and therapeutics. Together with the combination of our AI-driven workflows, the potential impacts stemming from simulations at this scale and level of complexity are just now being discovered. Our simulations also inspired the development of US and international simulation data sharing hubs, the NSF-MULSI effort being one of the central of those. Our data have been shared with thousands of people worldwide, including academic and industrial groups. Scientists are using our data to find vulnerable areas of the spike protein for vaccine design and hidden druggable pockets for those pursuing drug development. This is also an important point. These simulations utilize a lot of HPC resources, but we consider them as an investment that yields returns in their use by many other groups. In other words, the cost of this work in terms of node hours is amortized across all the thousands of scientists who will continue to use our data. In addition to the science highlights I just discussed, there's also a number of HPC innovations that set this work apart. The publications, the journal covers, the large media coverage, and the data sharing, and then coupled with the HPC innovations. We showed 10 to 100x factor speedups for weighted ensemble simulations and the DeepDriveMD workflows. We showed excellent scaling of NAMD and VMD to nearly the entire Summit machine. We also, our workflow set a total number of flops exceeding 2 zettaflops in just a very short amount of time. And we set several high watermark achievements, including for ensemble pathway simulations, for adversarial autoencoded multiscale workflows, as well as for the simulation, the all-atom simulation of viruses. And what's so amazing is this was just 8 months. It's really just the beginning. Stepping back, thinking about all that we've accomplished in these 8 months, it is like climbing a mountain. Sometimes it feels like you're never going to make it, but then you get to that summit and you look out and just, wow, what a view. Seeing things for the first time, discovering things that no one before you ever even imagined. That's really what this is all about. And looking out from this new vantage point, it's so much easier to imagine a bigger, better, and brighter future for HPC and the world. Thank you.